Okay, we're moving on to lesson 4 in unit 1. And so in this lesson, we're going to talk about mutations. So let me share my screen with you. Okay, so in video 3, we learned about protein synthesis, and we learned that protein synthesis is when the genetic code that is in the DNA is transcribed onto messenger RNA, and then messenger RNA goes out to the ribosome, and that's where translation occurs and the protein is made. But sometimes the DNA gets that process of when DNA is replicated or when DNA is transcribed, when the A's go with the T's, the C's go with the G's, the A's go with the U's, and so on. Sometimes in that process, there are errors made. And this is what causes a mutation. So there are 3 basic types of mutations that can occur, and we'll talk about what causes mutations in a minute. But first, let's just look at what happens with the 3 different types of mutations. So you'll see right here at the top, we have the original copy of a gene sequence. So what this is showing us is the DNA sequence for one particular gene. And so you see you've got TAC, which stands for thymine, adenine, cytosine, guanine, cytosine, thymine, guanine, adenine, cytosine. So this is the original DNA strand, and these are, of course, your nitrogenous bases. And so if an error occurs or a mutation, one of 3 things that might happen is called a substitution. So in a substitution, the wrong nitrogenous base is put in place of the correct nitrogenous base. So if you'll look here, we've got TAC, and instead of the T, we have an A put into place. So everything else is the same. You've got AC, GC, TG, AC, and you got the same thing, AC, GC, TG, AC. All the rest are the same, but instead of the T, we have an A. And so that's called a substitution. Sometimes substitutions don't cause major problems because you just have that one nitrogenous base that's wrong. And we saw with the amino acid, let's see, with the amino acid chart here from our previous lesson, we saw that there's different codons that all code for the same amino acid. So you can see here that UCU, UCC, UCA, and UCG all code for serine. So if there's just one nitrogenous base that's mixed up, it doesn't always cause the wrong amino acid to be put in the protein. Okay, but sometimes it does. So it just depends. So a substitution is just when one nitrogenous base is messed up, okay? And then now an insertion, on the other hand, can cause lots of problems. So let's see what happens here. So here's your original strand, and you can see that in the original we have TAC, but with the insertion, there's an extra T added. Okay, so everything after that is shifted. It's shifted over to the right. And so that could cause everything after that to be messed up. Because remember, every 3 letters is a codon for a particular amino acid. So In the original, we needed TAC. And so if we look at TAC, that's not going to be the same as TTA, right? So instead of TAC, GCT, it's going to be TTA, CGC, and then TGAC. So this first codon is going to be wrong. It's going to be for the wrong amino acid. Okay. And then for a deletion, you see this is the original. And with a deletion, one of your nitrogenous bases is dropped off. So here we've got the TAC, but instead there's no T. So the T is dropped and it just goes straight to ACG. So again, we're going to have a big problem there because everything has been shifted. Instead of shifting to the right, like in an insertion, in a deletion, everything shifts to the left. Okay, and so that causes your codons to be read. Remember, your codons are every 3 letters, and so they get read incorrectly, and you have the wrong amino acids. And if the amino acids are put together wrong, then the protein won't work correctly. So let's, so the 3 major types of mutations are substitutions, insertions, and deletions. And so the description of each of them is right up here. So you can read over that again. And then there are 3 major causes of mutations. So the first one is error during DNA replication. So as we saw those up here, and sometimes just a random error occurs, no particular reason, and that can cause a mutation. Other things that cause mutations are mutagens. And so examples would be like X-rays. Mutagens are things that cause mutations. So X-rays, radiation from the sun, harmful chemicals, and pollutants. And then the third thing that can cause mutations are viruses. Okay. And then the thing about mutations is they are not always bad. So mutation, that word kind of makes you think, oh, that's bad. But mutations always harmful? No, they're not always harmful. Sometimes they are actually beneficial. So mutations can be beneficial if whatever that trait is that happened as a result of the mutation, if it gives that organism an advantage in surviving, then it can be beneficial. And then sometimes mutations are neutral. They're neither harmful nor beneficial. And so, and mutations occur quite often. In fact, most of us have several mutations in our DNA somewhere in our body. And usually those mutations are just neutral. Sometimes they don't even show up. So just remember, don't get it in your head that mutations are always bad. Sometimes they're good and sometimes they don't even matter. Okay, so next we'll talk about proteins, different types of proteins. And so that's the end of this lesson.